Today we are going to go over the cytoskeleton. To begin, we have three main components to make it easy. We have the microtubules, microfilaments, and intermediate filaments. Now these are spread out all across the entire cell. However, below I have drawn three different diagrams of a cell just to show which one is spread out for each cell individually. Notice how in the microfilaments I lined all across the cell membrane border. The purpose of this is to show that they were flexible and they play a crucial role in cell movement and cell division. Now if we draw the microfilament down below, what we see is that it has a very small diameter, and in respect to the intermediate filaments and microtubules, it has the smallest diameter compared to both of them. The thing about this is that it has double helix of actin. To understand the actin filaments, I'm going to jump over to muscles. As I have drawn down below, we have actin surrounding the myosin, so we can see how it differs from when it's relaxed and contracted in muscles. Similarly, in animal cells, when they undergo cleavage, what happens is a contractile ring is formed to cause a cleavage furrow to pinch the cytoplasm. You can see the ring down below. Going back to the cytoplasmic movement, cyclosis heavily relies on this actin-based filaments. These filaments can almost act as tracts for which vesicles and organelles can travel through the cytoplasm on. What's interesting about the microfilament is that it polymerizes and depolymerizes rapidly. Next we have intermediate filaments, which are intermediate in diameter. So it's made up of different types of proteins, but the main one for the DAT we should focus on is keratin. Notice how for the intermediate filaments, I just put them all over the cell. Well, visualize a pouch and a nucleus being inside the pouch. So it's almost like the intermediate filaments are holding it up. And the purpose of this visualization is to show that intermediate filaments create a nuclear lamina. So imagine the nuclear lamina being the pouch. This basically supports the nucleus and its activities. So the main takeaway here is it provides structure and support. Moving on to microtubules, they also provide structural support. However, notice how differently I drew the microtubules. I did it for this purpose to show that it has to do a lot with cell division as well as cilia and flagellal production. Since microtubules are just so messy, they need something called MTOCs, which are microtubule organizing centers. And these are in eukaryotic cells, and they are known as centrosomes. In fungi, they're just known as MTOCs. If you recall from cellular division, such like mitosis, we have the centrosomes and the microtubules connecting. So essentially, the microtubules organizing themselves during mitosis just means that it's the spindle apparatus. So in that specific situation, microtubules are the spindle apparatus. In chromosomal division or karyokinesis, karyokinesis is just the splitting of the nucleus, karyo meaning nucleus, not to be confused with cytokinesis, which is the splitting of the cytoplasm, the cleavage. Now moving on towards the centrioles, we have proteins surrounding the centrioles, which is just called the pericentriolar material. This material is useful because it helps make up the microtubules, which is known as microtubule nucleation. To specify, when we have two centrioles, we have a mother and a daughter centriole that are positioned 90 degrees from each other. Each of these is a hollow cylinder made up of 9 triplets of microtubules, so basically 27 microtubules, and we can see the structure below. This is called a 9 times 3 array. So if you look at this circle, if we draw some lines, it can help us visualize the actual centrioles. These centrioles would usually replicate during the S phase. Now, if you recall that I mentioned that microtubules aid in the production of cilia and flagella, well, if we have the mother centriole attached to the cell membrane, that causes a 9+2 array for both cilia and flagella. 9 times 2 array. So essentially, the main takeaway here is that microfilaments are for movement, intermediate filaments are for cell structure, microtubule are for cell structure, division, flagella, and cilia production. I hope this helps.